Huh?
Let's open our books to 12.1. Hold on, I gotta get my book.
No, right.
The more you struggle, the more it's
It's gonna be like the most.
just take it. Meredith, too.
Hey,
put it on me.
12.1.
Can I wake you up?
What page is that?
Yeah.
I'm gonna
326.
go to you and give me a thumbs up. Okay. I'm gonna tell you what, not right now.
Okay, listen up. 326, please.
I'm ready.
Bennett, Graham, Tucker. 326 in your book. Taylor, you on it? 326. Okay. DNA has a double helical structure. It's made up of these small individual units. What do we call a single individual unit?
Nucleotide.
Nucleotide. A nucleotide has 3 parts. They are the
Phosphate group, sugar, and nitrogen base.
sugar, phosphate, nitrogen base. Very good. What are the 4 types of nitrogen bases? Adenine,
Guanine.
guanine,
Thymine. Oh.
cytosine, and thymine.
G and C go together, and T and A.
G and C go together.
Grilled cheese.
A and G go together.
And A and T.
G and A are called
Purine base.
purines. C and T are called
It's because he'll from Connecticut or
Thumbs up.
Now.
See a little mixture? So it's like, oh, wait,
My
I didn't
turn.
do that.
My turn. Now, the DNA holds the instructions. The instructions for making cells. And we're going to learn how it works later this chapter. It was first realized that there must be some... It was first realized that there must be some substance that transforms, that passes on information in this famous experiment that you need to know.
Mouse.
Could y'all stop, please? Whoever's humming. This isn't easy to understand, so hang with me. There's a scientist named Griffith who took some bacteria that he called S-strain bacteria. And he called them S-strain because they were smooth. They had a smooth outer coating on their bodies. And when he injected these smooth S-strain bacteria into a mouse, the mouse died. So, S-strain bacteria injected into the mouse, and the mouse dies.
Wait. S-strain?
They're called S-strain.
Oh, S-strain.
Who's making noises here? Who is that?
What do you mean? I was talking.
Besides you, there was somebody making noises.
Wasn't me.
Some humming or something.
I hear it.
It's going to be a long time we'll be in here explaining this one picture if we can't move on. Okay? So, what was your question, Todd?
I was just wondering. I didn't hear you right when you said.
They call this bacteria S-strain because the outside of the bacteria are smooth. The bacteria produce a little a covering, a coating on their bodies that are smooth. And if you inject those into a mouse, the mouse dies. Now, there's a different kind of bacteria called R-strain bacteria that do not produce the smooth outer coating. And if you inject those into a mouse, the mouse lives.
Does it change the mouse in any way?
No.
The mouse is unaffected.
These end up killing the mouse. These end up not killing the mouse.
What?
Take the S strain bacteria and heat it in a test tube so it dies, and then inject that into the mouse, and the mouse lives. So this kills the mouse, but if you heat it up, the mouse lives, and if you inject this R strain into the mouse lives. So both of these, this one and this one, don't kill the mouse. But if we take heat-killed S strain bacteria and R strain bacteria, put them together in a test tube and inject them into the mouse dies.
Why?
I'm going to get to that. Yes?
Are you heating the R strain
No.
also?
No. You're only heating the S strain. So this injected into a mouse doesn't kill them. This injected into a mouse doesn't kill them. But the two together into a mouse does kill them. So what in the world is happening? Well, yes, question?
Did they heat up the R strain?
They did not. That's what they just.
I know.
They did not heat the R.
Picture. What did they ever find out if they heated up?
No, they didn't heat up the R strain.
Okay.
Okay.
They said ever.
Yeah, no, I'm not talking about this picture in general. I'm talking about.
Like, what would happen if they heated up the R strain?
That's kind of what I'm trying to ask.
Well, the R strain by itself doesn't kill the mouse, so if you heated up the R strain to kill the R strain bacteria, that wouldn't kill the mouse either. So what was happening here? Well, they took blood from the mouse, and they looked at the mouse's blood, and they found living bacteria inside the mouse. And you know what it was? It was this. It was living S strain bacteria. Not heat-killed S strain bacteria, living S strain bacteria. So how is it that you inject S strain bacteria that's dead into the mouse, and if you do it along with R strain bacteria, it somehow comes back to life is what it looked like.
It grows in the R.
Yes, tough.
It grows on the R.
It what?
It grows on the R.
What happened was the R strain bacteria.
That's good.
What happens is the R strain bacteria takes information from the dead S strain bacteria. And that information allows it to make this smooth outer coat. So it's really the R strain bacteria that use some information from here to make a smooth outer coat, and thus are able to, they've transformed themselves into S strain.
Is it cooler to be an S strain?
You can kill mice if you're an S strain bacteria. I don't know if that's cool or not. Later, scientists in investigating this, they found that the substance that the R strain bacteria were absorbing from the S strain bacteria was DNA. They found the R strain bacteria would pull DNA out of the bodies of the dead S strain bacteria and use that information to make these smooth outer coats.
Cool.
That was a major experiment done back in the 30s that helped show that there was some information material that was being transformed from one bacteria to another. Here's another one that you need to know.
Are these in the book?
Yes, page 328. This one's on 328.
Pills?
Now try and follow this one.
Oh, 28.
328?
Gotcha.
Okay, y'all with me? These aren't easy to understand, so you got to hang with me here. Drew.
Sorry, I'm
Stop.
blind.
Rude.
So not okay.
No, it's not okay. Here we are growing bacteria, E. coli bacteria, in a Erlenmeyer flask. And scientists long knew that viruses somehow transferred information to cells. Scientists figured this out a while back. Because a virus will land on a cell, And the cell will then produce more viruses. So how does the cell know to produce more viruses? Information must have passed from the original virus into the cell. So this was an experiment to figure out what that information was. Scientists knew that viruses were made of two things. They were made of protein and they were made of DNA. Which one was being passed into these E. coli? What would happen is they'd mix the virus in the E. coli, and the E. coli would make more viruses. So what they did was they took the virus, and one group of viruses, they labeled the DNA with P32. P32 is a radioactive form of phosphorus. So what they did in this virus, they made the DNA glow. They made its DNA glow.
Cool.
In this virus, they gave it a radioactive form of sulfur, sulfur-35. So they made its protein glow. So this set of viruses has glowing proteins, and this set of viruses has glowing DNA. And they put them in beakers, and they let the viruses attack the cells. And the viruses injected their DNA. We found out because of this experiment, the viruses inject their DNA, not inject their proteins. They inject their DNA. And they shook up the mixture in a blender to get the viruses off of the bacteria. And then they looked at the bacteria. The way they did that was they took the mixture and put it in a test tube and spun the test tube around. Have you ever seen a centrifuge? You know what that is?
Okay.
It's a machine that spins things around real fast. And if you take a test tube full of bacteria and viruses and you spin it around real fast, the heavy bacteria go to the bottom of the test tube, and the light viruses will kind of stay up in the mixture. And so they spin it around. The heavy bacteria are on the bottom here, and the light viruses are up on the top. And then they check to see what's glowing. Now in this one, where the radioactive DNA was, the cells were glowing. They were glowing radioactive down here. What does that mean? It means the viruses transferred DNA into the cells. Over here, the proteins were radioactive. The viruses didn't stick the proteins into the cell. So the radioactivity was coming from up here, not from the cells down in the bottom of the test tube. So this proved that DNA was being passed from viruses to bacteria and not proteins being passed from viruses to bacteria. DNA was carrying the information from the viruses to the cells. You want to see video footage?
Sure. Pro.
That's hard to understand. Those two experiments are hard to understand. When you do your reading, if you didn't understand what I was talking about here, do your reading and try to figure it out.
Correct.
So viruses carry DNA to the cell?
Viruses pass DNA along into cells.
Correct.
That's how viruses work.
I understand the whole DNA actually passed through because the protein really isn't holding any information.
That's right.
But I don't understand like when they spun it. I don't understand that.
Well, bacteria are heavier than viruses. So if you have a test tube full of bacteria and viruses all mixed together, and you spin it around real fast,
Okay.
the bacteria go to the bottom of the test tube
Okay.
because they're heavier.
Okay.
And the viruses kind of stay up in the mixture. So it's a way to separate the bacteria and the viruses.
Okay. So if the virus had infected a lot of the bacteria, there'd now be more viruses.
Well, what they did was they had the viruses infect the bacteria. Then they blended them up in a blender so the viruses came off of the bacteria.
Okay.
And then they separated them by centrifuge.
Okay.
Does that make sense?
Not really, but okay.
Let me show you this video and
Okay.
you tell me. Tell me if this helps.
Your camera's about to die.
Okay, that's okay.
That was a good one, Drew.
Corn dogs. That's what it looks like.
Pretty sure it's bacteria.
Take a look at this. Viruses injecting radioactive DNA into cells. Put them in a blender and the viruses come off of the cells.
Okay.
Put them in a centrifuge and spin them around. The heavy cells go to the bottom of the mixture. The light viruses stay on top.
Okay.
The heavy cells are glowing radiation because the viruses pass their radioactivity into the cells.
Gotcha.
Got it now?
Gotcha.
It's like if you've got
Okay,
a
video footage.
phone and a bug on your phone. Like your phone, it flies off. True. Enable.
1952. Big cars and modern appliances. With World War II behind them, scientists applied their wartime technology to peaceful uses, including their quest to find the genetic material of life. Although challenged by some, many scientists believe that proteins, not DNA, held the secrets of heredity. Then two microbiologists, Alfred Hershey and Martha Chase, made an extraordinary discovery. Using radioactive isotopes, they traced the DNA of certain bacterial viruses. Ironically, using an ordinary household blender to separate the viruses from their bacterial hosts.
We tried various things that didn't work until we got this elegant little machine, which was invented, I think, to make cocktails. It worked right away. It worked fine.
In this pivotal experiment, Hershey and Chase proved that the genetic material of the virus was not a protein, it was DNA.
That didn't show the experiment. This shows the experiment.
Enable.
Okay, see if you can follow this. Let's see if it even opens up.
In the Hershey-Chase experiment, bacterial viruses, called phage, were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria.
Now you see what's happening here. Let's look right here. The red stuff these viruses contain, that's radioactive DNA. And here what they do is they squirt it into the cell. Now it's in the cell. Now they shake the cell and these coats fall off.
Yeah. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
See, this cell will now glow radioactive. If you shake them up in a centrifuge, which I'll show you, we got one in here in the other room. If you shake them up in a centrifuge, you'll be able to see the glowing bacteria at the bottom of the centrifuge.
So from then on, will all of the DNA be glowing?
Yeah.
How do you color the DNA?
How do you what?
How do you make the DNA color? Radioactive.
You
inject it, right?
Yeah, you have to plant radioactive isotopes of phosphorus. Remember a sugar, phosphate, and nitrogen base?
Yeah.
They make the phosphorus part radioactive.
So then, like,
And it's hard to do. I mean, it takes some chemicals.
so was all like the new phage that was made? It was glowing. Like
That's correct.
It had that altered.
That's correct.
DNA
That's
in it.
correct. The ones that it was making inside of it.
Okay.
Yeah.
Okay.
We'll talk about how a virus works.
Pro, you gotta say.
Bring your turn that off.
You gotta say Wilson.
Bring your sheets, your microscope sheets from last time into here, and I want to show you a centrifuge.
Willis out.